Cyclic AMP is a second messenger. It's released into the cytoplasm in response to the first messenger binding to a receptor on the cell surface. There are a number of other second messengers in eukaryotic cells, among them calcium, diacylglycerol, inositol triphosphate, cyclic GMP. And second messengers as a class can activate many cell activities, leading to large-scale and coordinated responses. So second messengers are going to accumulate in cells, or in some cases be inhibited within a cell, but the second messenger serves as an important signaling molecule. We've kind of talked about second messengers already. Here we're going to kind of talk about what happens as they accumulate, or as they fall within the cell. Here's an example of an agonist binding to a G protein coupled receptor in the upper left panel. And this receptor is not drawn to show you its 7 transmembrane spanning domains and such, but what you do see is the activated G protein affecting the effector enzyme, stimulating it, the effector enzyme being adenylyl cyclase, and cyclic AMP in that rectangle accumulating within the cell from the ATP. So the adenylyl cyclase takes the ATP and cleaves off the 2-phosphase and turns it into a cyclic AMP compound. Remember, cyclic compound, which is cyclic because of the phosphodiester bond that it contains. Now, if you were to then look at why that's important, it's important because there's an enzyme within cells that can become active in the presence of high levels of cyclic AMP. The enzyme is depicted for you at the bottom center portion of the slide. It's called cyclic AMP dependent protein kinase. Also it's called CAMP kinase. Also it's called protein kinase A. And this kinase is made up of 2 regulatory subunits and 2 catalytic subunits. The catalytic subunits are what is going to phosphorylate things, but normally the catalytic subunits are inhibited by binding to the regulatory subunits. So, under conditions when cyclic AMP levels accumulate, they bind to the regulatory subunits and pull the regulatory subunits off of the catalytic subunits. The catalytic subunits are now free to phosphorylate substrates, and those phosphorylations lead to cellular responses. Okay, so again, the cyclic AMP, as it accumulates in the cell, the second messenger goes up, the regulatory subunits are impacted, they fall off the catalytic subunits phosphorylate the substrate, leading to a cellular response. What is the cellular response? Well, as we've mentioned, phosphorylation is a dramatic kind of change in cells, and so that cellular response might be activation of an enzyme, might be inhibition of an enzyme, it might be protein synthesis, it might be nerve stimulation, it might be muscle relaxation. It's going to depend on the tissue, and so one example of the consequence of substrate phosphorylation is discussed next. Well, no, I lied. I'll discuss it in the slide after this. So, in this slide, the focus of this slide is what happens when the agonist falls off this kind of receptor. What happens when the agonist falls off this kind of receptor? We've just seen how accumulation of cyclic AMP is critical in terms of cell signaling, and we're going to get back to that in the next slide. But here, when the agonist falls off the receptor, the GTP eventually gets hydrolyzed on the G protein, right? So step 1 is GTP hydrolysis. When the GTP is hydrolyzed, the G protein stops stimulating the effector enzyme adenylyl cyclase. And so you won't get further formation of cyclic AMP. However, if you look at step 2, there's a second step involved, because you have made a lot of this cyclic compound, which is otherwise stable. Right? So if you want the signal to stop, you got to be able to turn that off. Right? You got to be able to degrade that. And so step 2 is inactivation by a class of enzyme called cyclic nucleotide phosphodiesterases. Cyclic nucleotide phosphodiesterases. And these are ACEs, enzymes, that cleave phosphodiester bonds. So the cyclic compound becomes a non-cyclic compound. And so the CAMP, or the cyclic AMP, is not around to bind to the regulatory subunits. Right? So the regulatory subunits bind back to the catalytic subunits, and the tetramer, the tetrameric protein kinase A, comes back together, but is not functional. Right? It's off. And while that seems to be a interesting way of inactivating everything, we still have substrates that have been phosphorylated due to that protein kinase A that's gone around and phosphorylated things. Step 3, therefore, is to cleave those phosphates off through the action of phosphatases, which will lead to those diminished cellular responses. So when the drug falls off of this type of receptor, several things start to happen to reset the tissue to respond again, right, and to turn off the pathways. So GTP gets hydrolyzed, cyclic nucleotide phosphodiesterases get stimulated, and phosphatases are activated to cleave those phosphorylated substrates. If we look at cyclic nucleotide phosphodiesterases, they're an interesting target for certain drugs, including drugs of the methylxanthine class. There are many types of methylxanthines, but the two most prominent members are caffeine and theophylline, which are present in tea. And these types of methylxanthines block cyclic nucleotide phosphodiesterase, and so cyclic AMP levels stay high. And so in the brain, for example, high levels of cyclic AMP are correlated with an ability to focus better, to concentrate, to wakefulness. Maybe that's the best term, wakefulness. So what's the main reason why so many of us make sure we get a daily dose of caffeine through our coffee or our Starbucks? It's because caffeine is going to cause levels of cyclic AMP in the brain to stay high, which is going to contribute to wakefulness. Right? Not just caffeine, theophylline, other types of experimental methylxanthines, synthetic methylxanthines can do these kinds of things. So, what do you expect the levels of caffeine to be on cyclic AMP? Will the levels be higher or lower than they otherwise would in the presence of caffeine? And how about the effect of caffeine on CAMP kinase? Would CAMP kinase be more active or less active in the presence of caffeine? I won't answer those questions, but hopefully the answers are obvious and they're easy to find in your book. If it's still unclear, or you can ask me. So now back to phosphorylation and the consequence of phosphorylation by CAMP kinase, or PKA. So different cells express different types of substrates for CAMP kinase, which can help explain some of the tissue-specific effects. So for example, in the liver, there's an enzyme called phosphorylase kinase that is activated by PKA, also known as CAMP kinase. So phosphorylation of that kinase activates it, and it's going to break down glycogen into glucose molecules. On the other hand, the enzyme glycogen synthase in the liver, when it gets phosphorylated by CAMP kinase, is inhibited. And so phosphorylation of glycogen synthase is an inhibiting response. Phosphorylation of phosphorylase kinase is a stimulating response. Phosphorylation can stimulate, phosphorylation can inhibit. It depends on the enzyme, it depends on the tissue. But here it makes sense, because if we're going to release glycogen, release glucose from glycogen, then would we want to just build glycogen right back? It wouldn't make sense, right? So in the upper pathway, if glucose is being released from glycogen, glycogen is being broken down, then the body needs glucose. So it's not a good time to store glucose, which is what glycogen synthase does. So by its phosphorylation, it's blocked. So here's one kinase activated by cyclic AMP, high cyclic AMP in the cell. One enzyme is phosphorylated and stimulated. One enzyme is phosphorylated and blocked. And so you can have, a whole plethora of cell responses coming out of this type of wiring of second messenger being cyclic AMP. The next system we'll discuss is calcium and phosphoinositides. And this is a well-studied second messenger system as well. It is also going to be used by a G protein coupled receptor, but this one is going to be coupled to the GQ receptor. And there are many examples of GQ coupled receptors in the body. One of them is one type of muscarinic receptor. The M2 receptor. The M1 and the M3 receptor are GQ coupled. The M1 and the M3 receptor, which we're going to kind of dissect more in a later chapter. So M1 and M3 are GQ coupled. The alpha 1 receptor in the vascular smooth muscle GQ coupled, and then there are many other examples, but those will be two receptors we talk about in detail later. And again, the second messenger here are calcium and phosphoinositides. And those phosphoinositides are things like diacylglycerol, DAG, and inositol triphosphate, IP3. And so in this situation, the drug binds to the receptor, the G protein becomes active, but this is a GQ-coupled receptor, and so its effector enzyme is an enzyme in the membrane called phospholipase C, lipase ACE that cleaves lipids. What kind of lipids? Phospholipids. The substrate for PLC is called PIP2, which stands for phosphatidyl inositol 4,5-bis-phosphate, or just simply put PIP2. And it gets cleaved by phospholipase C into two second messengers. One of them is called inositol triphosphate. The other is called diacylglycerol. The inositol triphosphate comes into the cytoplasm. The diacylglycerol stays at the membrane. Okay, but neither of them would exist if phospholipase C hadn't done something, and phospholipase C couldn't do something unless it had been stimulated to do something by GQ. The IP3 will bind to receptors on the endoplasmic reticulum within target cells. And the endoplasmic reticulum is an important calcium sequestering organelle. We've learned about the endoplasmic reticulum in terms of the smooth ER and the rough ER, and you've probably learned about the rough ER and protein synthesis through translation. But here, we're looking at the ER as a calcium storage site. Right? Calcium is a very important ion. To give you an idea how important it is, there's a journal in the library called Calcium. And it's been in publication probably for 50 years or maybe more. Okay? So every month people publish on this ion, right, what it does and how important it is. So it's a very important ion. So IP3 accumulates in the cell, binds to receptors on the surface of the ER, and then the ER dumps the calcium. You see, the IP3 binds to the receptors on the ER dumps the calcium. Now the calcium, when it gets released into the cytosol, binds to a protein that you see here at the very bottom called calmodulin. And that's a protein whose activity is modulated by calcium. So calcium will bind to it, and watch what happens. It goes from being kind of an inhibited protein to being a stimulated protein in the next slide. And so that calcium calmodulin, and it's 2 calciums for every protein of calmodulin, for every cofactor. That calcium calmodulin complex will then activate a kinase called calcium calmodulin dependent protein kinase, as you can see in the red box. And that's going to happen like what we see in the next slide. And so the calcium calmodulin complex stimulates the calcium calmodulin dependent protein kinase. And we start to see substrate phosphorylation. Now it seems like a lot of words, right? And maybe it's overwhelming, but really if you look at the words, they're not that bad, right? They're just talking about what is needed to activate the enzyme, calcium and calmodulin, the calcium-calmodulin dependent protein kinase. And now we start to see substrate phosphorylation. That calcium-calmodulin stimulates the calcium-calmodulin dependent protein kinase, and so substrates get phosphorylated, and that can result in cellular responses. If we're in the vascular smooth muscle, the cellular response may be constriction of the blood vessel, right? Contraction of that smooth muscle, for example. Now, we've talked about the IP3 and what's happening as a consequence of its accumulation. But we haven't yet talked about the diacylglycerol. The diacylglycerol is another member of this second messenger family. Really, there's three: IP3, DAG, and calcium. The diacylglycerol will actually activate a protein kinase inside the cell that you see here in the middle portion of the slide in a red box. It says PKC inactive. That stands for protein kinase C inactive. Protein kinase C will become active when it interacts with DAG. And DAG will only be around to interact if PIP2 has been cleaved. And so watch what happens in the next slide. The protein kinase C becomes stimulated by the DAG. And in the presence of calcium, the C, by the way, stands for calcium. In the presence of calcium, the protein kinase C will phosphorylate substrates. Okay, so protein kinase A is referring to CAMP kinase. Cyclic AMP level has to be high to activate protein kinase A. In protein kinase C, the calcium level has to be high in the cytoplasm, and there has to be an interaction with DAG as well. And so phosphorylation of substrates by protein kinase C will cause cellular responses. Now, in addition to like general, regular old calcium-calmodulin dependent protein kinases that can phosphorylate a wide variety of substrates in cells, there are certain specialized calcium-calmodulin dependent protein kinases that can, for example, if we're in muscle, skeletal muscle, affect the myosin light chain kinase, for example. Right, so this can become more complex than what I've presented to you. So there are, in addition to just the regular old calcium-calmodulin dependent protein kinase, there can be specialized ones in certain targets. And in terms of protein kinase C, there have been at least 9 different types that have been identified. Right, so these pathways can quickly become complex, but the simplified mechanism is a GQ-coupled receptor becomes stimulated by an agonist. GQ activates the effector, phospholipase C, and then things like IP3, DAG, and calcium go up, and cell responses happen. And so just like we talked about, what happens when, when an agonist falls off of a receptor that was coupled to GS that turned on all this cyclic AMP. What happens when an agonist falls off a receptor that's coupled to GQ? How does that stop signaling? Well, first of all, the IP3 gets dephosphorylated by phosphatases, so it's not IP3 anymore. And the diacylglycerol is metabolized. It's either phosphorylated and converted back into a phospholipid, or it's deacylated. But whatever the case may be, it's not DAG anymore. So IP3 gets altered by phosphatases, DAG gets altered, and the calcium that was dumped into the cytoplasm gets actively removed from the cytoplasm by calcium ion pumps back into the endoplasmic reticulum. And so all of these elements of the calcium signaling pathway are becoming potential targets for new drug development. Cyclic GMP, like its counterpart cyclic AMP, is a cyclic nucleotide. Right? It's a cyclic guanosine monophosphate And it's cyclic because there's a phosphodiester bond that's making it a circle type of cyclic compound. But unlike cyclic AMP, cyclic GMP has really only been identified as a second messenger in a handful of cell types. Among these are the intestinal mucosal cells and the vascular smooth muscle. And so in these cases, ligand binds to the extracellular domain of the receptor. The ligand binding stimulates an intracellular guanylyl cyclase activity. The cyclic GMP that starts to accumulate will activate a kinase called cyclic GMP-dependent protein kinase. So that's depicted for you here in the bottom with this red cyclic GMP-dependent protein kinase becoming a stimulated green on an active cyclic GMP-dependent protein kinase. Substrates get phosphorylated. And so we've talked about cyclic GMP, for example, in smooth muscle as being relaxing. So when those levels are high, we get relaxation. And activation of cyclic GMP-dependent protein kinase leads to that relaxation of smooth muscle. Okay, so you'll see cyclic GMP, cyclic GMP, as a second messenger in a few tissues, the intestinal mucosa, the vascular smooth muscle, the eye, but it's definitely not as ubiquitous of a second messenger as, for example, calcium or cyclic AMP is. And so just to remind you of how we can get increases in cyclic GMP, we had talked a little bit about the atrial natriuretic factor that binds to its receptor, which is a receptor guanylyl cyclase. Cyclic GMP levels go up, which can cause blood vessels to relax. We had also talked about nitric oxide, and nitric oxide being able to enter a cell as a hydrophobic molecule and bind to a soluble form of guanylyl cyclase. So remember the difference between a soluble guanylyl cyclase or receptor guanylyl cyclase. And so that nitric oxide comes in, and guanylyl cyclase is stimulated. And so cyclic GMP levels go up, and we get relaxation of vascular smooth muscle. One easy way to remember that may be to think of nitric oxide as endothelial-derived relaxing factor. And so there are several vasodilator drugs that you will learn about in future course. We talk about the cardiovascular system, which work by mimicking nitric oxide.